Hello everybody, and welcome to your overview of microbial genetics. We're going to start off with what genetics and microbial genes are, what they're composed of, and how they replicate. So this is just going to serve as a mini lecture, and then we will go over some critical thinking examples in class. So as you have become accustomed to know, this is a microbial genome, okay? Circular, it can be kinked at places, and this is, of course, an electron micrograph. So the big pictures. Genetics, again, is the science of heredity. The central dogma of molecular biology is what we're going to go through, which is DNA goes to RNA, RNA goes to protein. However, as we start looking at viral life cycles, you'll notice that the central dogma is violated several times. We're going to go over mutations as well as how gene expression is controlled. So this is kind of an overview of some of the things that we're going to talk about today. This is the flow of genetic information. This is mutations, and these are control of genes, when which genes are expressed and when. So the type of events that we're talking about here, again, you have your DNA, and sometimes the DNA is replicated, okay? And then you have binary fission or mitosis, whatever organism you're talking about. But when we're talking about creating functional proteins, we're going to create DNA. We have DNA starting with it. Then we have messenger RNA. Now, I'm going to call this message. So if you hear me saying message, I'm just referring to messenger RNA. Now, again, DNA is very different from RNA in that it uses uracil, okay, instead of thymine. So it is deoxyribonucleic acid for DNA and ribonucleic acid for RNA. There is no proofreading function for RNA. So if you make an error in your RNA, you have proteins such as ubiquitin, which will just come around, recycle all the nucleotides, and then you can use them to make more RNA later. Then we have a protein, and the protein can be something like the keratin in your nails, or it can be a digestive enzyme in your stomach, or a metabolic enzyme from your liver. These proteins range in function and are basically the workhorse of the cell or body. Function is what the actual function is. Now, if you get a mutation in your DNA, and that mutation is not fixed by your DNA polymerases or any of the proofreading functions that you have, this leads to mutated RNA. And mutated RNA will lead to altered proteins. Now, sometimes this can be a good thing. So there are certain people in society that have mutations in their metabolic enzymes, so they are more efficient at getting rid of toxins or carcinogens or cancer-causing agents, or maybe they're more efficient at repairing their genome. So those mutants are good, but most often mutants are bad, cancer-causing in humans. But because microbes have such a high reproductive rate, they are going to lose many organisms to errors in mutation. However, the ones that produce good mutations are going to be around for a while. So when we're talking about mutations, there's several kinds. There is a base substitution. Now, a base substitution is when you have a change in a single pair. Now, these base substitutions or these frame shift mutations where a reading frame just gets kind of tweaked a little bit, these are referred to as single nucleotide polymorphisms. So single, one, nucleotide, right? These are nucleotides that make up our DNA. Polymorphism or mutation, a change. And we often refer to these as SNPs. So these SNPs can lead to actual functional mutations or be completely silent. So frame shift, again, DNA base pairs are added or removed from the sequence, causing the frame to shift, which causes the message to be read incorrectly. So that's mutations overview. This is our inducible operon versus our repressible. So you have to think about this in terms of what is the organism most likely going to need. So let's talk about this in terms of sugar. Lactose is a far more exotic sugar than glucose. Okay, so if something is rare in the environment, you're not going to want to be producing proteins regularly for that because it's a waste of resources. So this is when we have inducible. Inducible means it has to be turned on. So you have this thing called an active repressor. And this active repressor sits down on the gene and prevents that mRNA from being made. Now, an environmental stimulus or an inducer might come around and sit on that active repressor to remove it. This says, hey, guess what? This stuff is in the environment. Let's go ahead and start making it. Whereas we have a repressible operon. This thing is by default on. Okay, so this is always going to get coded. The protein is always going to get made until there is an environmental message that comes through that turns it off. So inducible, default is off. Repressible, default is on. So, big picture. Alteration of bacterial genes and gene expressions. They can cause diseases such as cancer. They can prevent disease treatment because maybe the genetic mutation requires you or makes you mutate at a higher rate, or causes issues with metabolism of the treatment itself, making the treatment ineffective. We often manipulate it for human benefit. For example, when we create vaccines, we alter the viruses or bacteria in such a way that they are unable to cause us disease, yet still look similar enough to the organism to prevent or to alert our immune system. Treatment of genetic diseases, we're starting to look at how we can use gene mutations and genetic information to knock out genes or at least turn them off. Now, as you go through your book, they're going to have examples up on the board right now are 3 different examples of different genetic advancements. I want to encourage you to kind of review these on your own. So, structure and function of genetic material. Genetics, again, the study of how they carry information, how information is expressed, and how genes are replicated. Whereas the chromosomes are structures containing DNA that physically carry the hereditary information, the chromosomes contain the genes. The genes themselves are the segments of the DNA that encode products, usually proteins. The genome is all the genetic information within a cell. Now, we talked about the structure and function. So we go from DNA to RNA to protein. So again, DNA, we have proofreading, so we can ensure that copies of our DNA are accurate. RNA, right, we have no proofreading function for RNA, and that leads to our protein. So genotype is the genetic makeup of the organism, whereas phenotype is the expression of the gene itself. So for example, my genotype could be that I have the dominant gene for freckles, right? And my phenotype would be the actual freckles themselves. A better example of this are things like my recessive red hair gene. So I did not know I had a recessive red hair gene. My wife did not know she had a recessive red hair gene. Yet one of the 3 of our children came out with bright iridescent red hair, which suggests our genotype was that of heterozygote, or a carrier, and then when we reproduced, we produced a child with that ginger gene. So bacteria usually have, again, a single circular chromosome made up of DNA. They have what we call short tandem repeats. These are repeating sequences of non-coding genes. Sometimes these short tandem repeats can be promoters or enhancers, which increase the amount of mRNA, increase the amount of mRNA that get made. When the more mRNA is made of a gene, more of that protein is made, or ideally, if environmental nutrition is appropriate, more of that protein is made. So here you have, a bacteria, and you have an electron microgram of how big the chromosome is that is packed inside that little nucleoid. So, vertical gene transfer will take us to our next bit of information, which is how does genetic information get from one generation to the next?